Hey guys, Mr. P. In this video we're going to talk about water movement specifically through plants, and this video is going to include 4 of the IB objectives, D2, 3, 8, 9, 10, and 11. The objectives specifically state, in the case of D2, 3, 8, is water potential as the potential energy of water per unit volume. Students should understand that it is impossible to measure the absolute quantity of the potential energy of water, so values relative to pure water at atmospheric pressure and 20 degrees C are used. The units are usually kilopascals or kPa. D239 says movement of water from higher to lower water energy. That's going to be higher water potential to lower water potential. Students should appreciate the reasons for this movement, and in this video we will talk about the reasons. In terms of potential energy, D2310 says contribution of solute potential and pressure potential to the water potential of cells with walls. We're going to use this particular equation, which is the potential energy of water equals the potential energy of the solute plus the potential energy of the pressure. Students should appreciate that solute potentials can range from 0 downwards, so they're always negative, and the pressure potential are generally positive, although negative pressure potentials occur in xylem vessels specifically where sap is being transported under tension. D2311 says water potential and water movement in plant cells. Students should be able to explain in terms of solute and pressure potentials the changes that occur when plant tissue is bathed in either hypotonic and hypertonic solutions. And this objective specifically is going to manifest itself in a lab experiment where we will actually investigate the effect of water potential looking at hypotonic and hypertonic solutions. So the first thing we need to do to kind of kickstart this particular video is we have to talk about what water potential is. Water potential, like the objective says, is the potential energy of water, which is what we're solving, equals the potential energy of solute plus the potential energy of the pressure. And so water potential is never positive, and that's because your solute concentration always is negative, and the higher the solute content, the more negative that particular variable is. The water potential of pure water at atmospheric pressure and 20 degrees C is 0. So that's kind of our standard. Water potential of pure water with no solutes added at atmospheric pressure and 20 degrees C is 0. Solutes attract water molecules and make it harder for them to move, so the water potential of a solute will always be negative, meaning the more solute you place into a solution, whether it's little solute or lots of solute, is going to cause the potential energy of water to become more and more and more negative. So the more and more hypertonic a solution gets or is, the lower or the more negative the water potential will be. And so if you think about that, just to reiterate that concept, solutes, because they attract water molecules, meaning these solute particles are actually going to attract and kind of interact and link themselves to these water molecules. It's going to impede the water molecules from moving. It's going to cause the water potential of those water molecules to drop. It gets harder for them to move, and so the more solute you have in a solution, the lower your water potential energy gets. And so when we kind of put all this together, the movement of water in a solution or among solutions or in and through cells is called osmosis, and osmosis can actually be defined using water potential. Osmosis is the passive movement of water particles from a region of high water potential to a region of low water potential through a partially permeable membrane. So if we talk about water moving from a high water potential through a membrane, and we can say a semi-permeable membrane, to an area where it has low water potential, that's how we're going to define osmosis. And this is the way that we would define the movement of water using water potential. But remember that in order to have a higher water potential, you have to have a lower solute potential, because the more concentrated the solute gets, the more negative the water potential gets, which means the water potential energy drops. So water is going to move from an area of high water potential to an area of low water potential, or it's going to move from an area of low solute potential to an area of high solute potential. Water will have a high water potential if there is a low solute concentration, and as this water moves from an area of that high water potential to an area of low water potential, it's going to eventually reach an isotonic point, or a point of isotonicity, meaning the water potential energy is going to be equal on both sides of the membrane. So again, if we have a semi-permeable membrane and the movement of water is going from high water potential to low water potential, it will eventually move until both sides equal. At that point, isotonicity is reached, which is basically a way of saying equilibrium is reached. And at that point, your net movement of water will stop. However, you will get movement in both directions, meaning one water molecule will move in one direction and another water molecule will move in the opposite direction. The net movement of water stops, but the total movement of water never stops. So, here is an example. 3 different situations. If we look at A, the solution on the right has a negative water potential due to the presence of the solute. So we have pure water on the left. Again, we have a semi-permeable membrane separating the 2 solutions. The volume on each side is exactly the same. The one on the right, or the solution on the right, has a 0.1 molar solution. It doesn't matter what the solute is, but because it has a 0.1 molar solution, we know the presence of the solute will decrease the water potential. And so you'll see over here on the left, the water potential is 0 megapascals. We also, because of the solute on the right, have a lower water potential at negative 0.4 megapascals. We know that movement of water will take place from a higher water potential to a lower water potential. Again, 0 is actually higher than negative 0.4. As the water moves to the right, it will cause the volume of water on the left to decrease. Notice the water used to be here. It has decreased. That amount of water has been added to the right side. It's increased the volume, so the volume has increased. And because the volume has increased, the concentration of the solute has dropped. We have a higher solute potential energy. Notice the solute potential energy started at negative 0.4. Because the solute concentration has been decreased with the addition of the extra volume of water, the solute potential energy has actually become less negative to a negative 0.15. But you'll also notice that because we added more volume, we have actually increased the pressure. Notice the pressure started at 0. That is the potential energy of the pressure. We've increased the pressure. So as the water moves from this left side through the semi-permeable membrane, every water molecule added is going to start increasing the pressure. It's also going to start diluting this solute concentration, which is actually going to make it less concentrated, and it's going to increase the potential energy of the solute, or the solute potential. And when we add pressure, it's going to cancel out, because pressure is positive. It's going to cancel out the negative solute potential, and it will eventually drop to 0, which is where our water potential energy is on our left. And you'll notice that on this left diagram, there is a one-way flow of water from higher water potential to lower water potential. But when the zeros are reached on both sides, meaning the water potential is the same on both sides, then the net movement of water stops, meaning water will move to the right and water will move to the left. If one water molecule goes to the right, there will be another water molecule go to the left. At this point, there is isotonicity reached. You have the exact same water potential energy on both sides, and they are said to be equal. Now, if we increase pressure on one side, if we look at C, the addition of this physical pressure will increase the water potential, and osmosis will continue until the solute potential exactly balances the pressure potential. Notice that we did not add volume. The left side of this diagram, in A, we started with a solute potential of negative 0.4. Over here, we still have that solute potential of negative 0.4. The only difference between all three of these diagrams is that we exerted pressure, which is increasing the positive of this variable, and when we balance or cancel out, exert enough pressure to equal our solute potential, we're going to actually reach a zero water potential, which is going to equate or be identical to our starting water potential, or the water potential on the left side. And so we're going to balance these sides, and just by exerting pressure on the right side, we'll actually reach an equilibrium point, and therefore the net movement of water is zero, and water can move back and forth through that semi-permeable membrane in equal proportions in both directions. So, an important depiction of how plant cells utilize turgor pressure to maintain isotonicity. The reason I put this in as a suitable diagram is that because plant cells have cell walls, and because their membranes are pressing against the inside of the cell wall, and because their central vacuole is filling with water and pressing from the inside out onto the cell wall, it increases that turgor pressure, which is exactly like what's happening on this C diagram. Keep that in mind as we start talking about what happens to the water potential in a plant as water moves through this water potential gradient. So the first thing to kind of key in on is that water potential decreases from the roots to the leaves in a plant. And so you can see that there are water potentials in various parts of this plant. Water in the soil is going to enter through the roots and will navigate from the roots through the root tissue into the xylem, which is part of the vascular bundle, part of the vascular tissue for a plant. And that water molecule, once it gets into the xylem of a plant, will make a single tube of water. Because water is polar, meaning it has both positive and negative poles, it has the capability of bonding to itself, and we call that cohesion. We've talked about the properties of water in previous videos, but water molecules can stick to each other, and they form these hydrogen bonds between the water molecules. The partial positive side of the water molecule, specifically the hydrogens, are going to have a partial positive charge, and the oxygen side is going to have a partial negative charge. The partial negative charge of a water is going to bond via a hydrogen bond to the partial positive side of a hydrogen ion, and it's going to create these hydrogen bonds. We call that cohesion. Now, water molecules can also create these hydrogen bonds between the vessel wall and themselves, and so we call that property adhesion. So through the properties of adhesion and cohesion, these water molecules will form not only a single chain of water, but they will actually use these properties to climb up from the roots, through the trunk, through the branches, and eventually will leave through the pores on the underneath side of a leaf, and those are called stoma. Water moves from a region of higher water potential to a region of lower water potential, and since the water potential is higher in the soil than it is in the roots or in the trunk, water will move from the soil to the roots. It will therefore move to the trunk. It continues to decrease, so all of the bottom, any part of the plant that is below a certain portion of the plant, water will always move to an area of lower water potential. And since water potential is decreasing as you increase up a plant or increase height, water will consistently move up to a region of lower and lower and lower and lower water potential before it evaporates out of the plant into the outside air. And again, because the air has a relatively low water concentration, it's going to have a very low, a very negative water potential. Water moves from the soil into the roots because there is a higher water potential in the soil than in the roots. Again, from the soil to the roots. Water usually moves from an area of lower solute concentration to an area of higher solute concentration, and solutes are concentrated in the plant cells. That's the reason the water potential continuously drops. Water potential is negatively impacted by solute concentration. So as the solute concentration rises, which it does, as you go up a plant, the solute concentrations typically get more concentrated. This is where all of the photosynthetic products are made. The solutes becoming more and more concentrated are going to cause the water potential to drop and go more negative, and so water will continually move up the plant that way. Transpiration is the process that involves the loss of water vapor through specialized plant structures called stomata, and most stomata occur in the leaves of a plant. And so this is where all or most of the water is being given off. And so as the water evaporates out, it's going to continually pull this single wide chain of water molecules up the plant as that water evaporates out of the top of the plant. Water evaporating from the stomata causes the pressure potential to drop in the cells of the leaves, and as the water potential then drops, additional water moves upwards. Again, as the water is evaporating out of the leaves, they're actually kind of creating a vacuum behind them, and that's causing the pressure even in those plant cells to drop. And if you remember that water potential equation, the potential energy of water equals the potential energy of the solute plus the potential energy of the pressure. Typically, remember, pressure is positive and the solute concentration is negative. However, as the water is pulled out, it creates kind of a negative vacuum, and it actually causes the pressure in the xylem to drop or go more negative, which even further decreases the water potential. If the solute concentration is increasing and the pressure is decreasing, that causes the potential energy of water to drop even further. So what's happening in a plant related to turgor pressure? So in a turgid state, meaning the plant has high turgor pressure, there is an equal water potential inside and outside of the plant. It is fully hydrated. In a low turgor pressure situation, the plant is wilting, meaning it has lost water. There typically is higher water potential outside of the plant than there is inside of the plant. And so there are two fluids we need to keep track of. You have extracellular fluid and you have intracellular fluid. The extracellular fluid is the fluid outside of a cell, and the intracellular fluid is the fluid inside of a cell. And so in a low turgor pressure situation, you have higher water potential outside than you have inside. You have higher pressure potential outside than you have inside, and you have higher osmotic potential outside than inside. Again, because the water in a cell has decreased, it has caused the central vacuole of your plant cell to shrink. When the central vacuole shrinks, it pulls the membrane away from the cell wall, creating this flaccid state, and there is less support from inside of the cell, causing the plant to not stand up correctly. In contrast to this extracellular fluid, you have lower water potential in the intracellular fluid. You have lower pressure potential because the plant membrane is kind of pulling away from that cell wall, and you have lower osmotic potential as well. So you can see in this time lapse, if you water a plant that has been wilted, you can actually see that it begins to take on water. Water moves into the plant, and it causes turgor pressure to increase until it actually stands up on its own. And in order to wrap all of this up, we'll do a quick example. So if I put a cell into a hypotonic environment, meaning the cell will be hypertonic in comparison to the environment. The cell is hypertonic, the environment is hypotonic. It means that the cell actually has a higher solute concentration than the outside environment. So the environment will have a lower solute concentration. If the cell has a higher solute concentration, the potential energy of the solute will actually be lower because it will be more negative, pulling the overall water potential down. So a hypertonic cell will actually have a lower water potential than the outside, which has a higher water potential because the solute potential isn't going to be as negative. If the environment has a higher water potential than inside of the cell, it's going to cause the water, which is in the environment, to increase the water volume of the inside of the cell, which is causing the water to go in. And as the water goes in, it's going to cause the membrane to expand and swell, and it's actually going to push on these cell walls, which will kind of bulge outward a little bit. And as the water moves in, it's not only increasing the amount of water inside of the cell, it's actually going to increase the pressure inside, which is going to cause that potential energy of the pressure to rise. And again, that is a positive number. As the pressure increases and the amount of water increases, it causes this water potential to increase, which is actually going to create a dynamic equilibrium with the environment and will cause them to be the same. And then if we contrast the hypotonic environment with a hypertonic environment, which means the solute concentration will actually be higher, which equates to a lower water potential. And so when a cell is placed in this kind of environment, that's going to equate to a hypotonic environment inside the cell, which means the water potential is higher inside the cell because the solute concentration is lower. And we know that water moves from an area of high water potential to an area of low water potential. And so the water inside the cell is going to leave because, again, it will be leaving from an area of high water potential to an area of low water potential. As the water pulls out or goes through the cell membrane will actually pull away from the inside of the cell wall. The volume of the cell will shrink, the pressure inside will shrink, and it will actually cause the cell to shrivel and could, in this case, cause the plant to wilt. And so as these cells are shrinking because of the loss of water, the plant will continually wilt and get droopy and lose its turgor pressure. And so the only way to ensure that your cells in a plant are maintaining turgor pressure is you want to either have them in a hypotonic environment or they need to be maintained within a certain degree of isotonicity, and so that there is a dynamic equilibrium reached, and so water can move in both directions equally. But for the most part, plants are typically kept in a hypotonic environment, and that's because the water potential of soil is higher than the water potential inside of the cell due to the high solute concentration of these plant cells. Water typically always moves in as long as water is constantly available to the plant. That's it for this particular video. If you learned something, give it a thumbs up, subscribe to the channel. We'll see ya.